have you here for one of our ICOPS workshops. And before I introduce our wonderful speaker for today, I just want to say, just so you're all aware, this workshop is being recorded and will eventually be edited and put up onto YouTube. So if you're uncomfortable having your face being shown on social media, just make sure that your camera is kept camera is kept off, but you're welcome to turn it on if you want. During the talk, please make sure that your audio is kept muted so that we can actually hear Dr. Ramsey speak. But then we'll open it up to questions and answers, and we're delighted to have all your questions answered during this talk. Javi is going to share a bit of information about the next speaker in the chat. Is that correct, Javi?
Yep.
Fantastic. And without any further ado, I am delighted to introduce Dr. Ramsey, who is going to be giving us the talk today, the workshop today. And I will just give a very brief introduction because I know it can be a little bit overwhelming to have a lot of information at the front. So Dr. Ramsey is currently am I right in saying assistant professor?
Yes.
Yeah, assistant professor at the University of Toronto. And she is leading the RLEA lab, the Ramsey Laboratory for Environmental Archaeology, which focuses on microbotanical analysis. So that's phytoliths, but also other microbotany like starch, the new science of starch spherulites, and microcharcoal. But importantly, in the context of this talk, she's been looking at the idea of how we can use AI to help with paleoethnobotany and how we can use it in this case in relation to phytolith identification. She works mostly in the area of southern Levant, but has also worked on the Canadian Plateau and is looking at questions around things like the Epipaleolithic, the pre-pottery Neolithic, and how hunter-gatherers and early cultivators are using plants in different ways for everything from food through to building materials, fuel, and other areas. And so we're really excited to have this talk about how AI might be useful for us as phytolith researchers and other people working in archaeobotany. So, Monica, I'll let you take it away.
Thank you for that very thorough introduction. I hope I can answer all your questions, but I suspect I won't be able to answer all of your questions. There's just so much more that needs to be done on this topic, but thank you for giving me the opportunity to speak with you all here today. So the research that I'm presenting here isn't just my research. I also have co-authors. So my grad students, Melanie Pugliese and Lachlan Kyle-Robinson, they are here with us today. And when we get to questions, they will also be answering questions. But also just my larger lab group, this research would not be possible without a team. It is truly a team science endeavor. So our talk is titled MicroBot, Developing AI Tools for Phytolith Identification. And I hope to introduce you all to our research aims, where we're currently at with this research, and then discuss with you some of the issues we're facing regarding open science in this new age of AI-assisted research. But let me just clarify some terms so that we're all on the same page first. So you have AI, which is artificial intelligence, and this is the overall field focused on building systems that perform tasks that require human intelligence. Then within that, you have the subset called machine learning, which is where systems learn patterns from data instead of being explicitly programmed. And then on top of that, you have a further subfield of machine learning, which is deep learning. And this uses multi-layered neural networks to learn complex representations directly from raw data. And then sort of surrounding deep learning and machine learning, it's sort of what can you focus your research towards? Well, we use computer vision and we use machine learning and deep learning to look and analyze visual data. And deep learning is sort of really powerful when it comes to vision learning. And who are we? So as Jennifer said, my name is Monica Ramsey. I'm an archaeobotanist and a microbotanical specialist. I did my research under Arlene Rosen at the University of Texas at Austin. I finished in 2015, and I've since held several postdocs. I was a terminal postdoc for quite a while at the University of Toronto and at the University of Cambridge before taking up my role here at the University of Toronto Mississauga in 2021. And the work, as I said, I'm talking about today is co-authored with my PhD students and my lab in general. And here we're building a lab group, a paleoethnobotany research group altogether, and that includes 5 graduate students right now, 28 undergraduates, and our undergraduates are sort of organized into teams, a paleoethnobotany team, a deep learning algorithm team, and an AI training team. And they come from a range of disciplines: anthropology, chemistry, biology, and a lot of computer science and stats students. We have a really talented group of undergraduates that we get to pull from. We're really lucky. In my lab, we're particularly focused on microbotanical analysis, but we're equally equipped to take on a broad multi-proxy approach to the archaeological records. So we've looked at food remains, for example, in my lab, and we can do macro materials for sure as well. But in addition to really developing AI tools for phytolith research, as Jennifer also said, we also have funding to look at and investigate the formation and application of starch spherulites to the archaeological record. So we're doing lots of cooking experiments and such in our lab right now. But for the purpose of today, one of our primary research aims in our lab is to develop a near to fully autonomous as possible, Phytolith AI identification workflow for the Southern Levant. We're a new lab, as I said. We established the lab in 2023. And with the help of my students, Melanie and Lachlan, we're really focused now on building the infrastructure that's going to allow us to scale up our research. Sorry, I'm just moving around slides here, or boxes here, so I can read my stuff as well. So you could ask, why develop AI tools for phytolith identification? And why was this an interest at all for someone who doesn't have a computer science background? I don't. Well, I like big questions. So my work up to this point has been focused on investigating epipaleolithic plant use in the Southern Levant. I've looked at sites like Ohalo 2 and Karana 4, and I want to look at and provide direct evidence for plant use in this 10,000 year period prior to the origins of agriculture. And I'm just frustrated by how long it takes. I want more than just 50 on-site samples to evidence 1,000 years of plant use at a particular site. I want off-site materials in particular. I want on-site material. I want data from every viable deposit so that I can discuss in detail this prolonged period of wild plant cultivation and the transition to the origins of agriculture. Accordingly, my focus when we started this endeavor was, how do we scale phytolith analysis? How do we scale it to hundreds or thousands of samples in a reasonable way? It wasn't necessarily to get better identifications or more specific IDs. For example, maybe the way to do that would be to develop SEM images. So use SEM training data from phytoliths if you want to see how specific we can get with IDs. I wanted to scale the methods that we're essentially already using. So phytolith extraction from sediments mounted on slides analyzed using good quality transmitted light microscopes. But in conducting our pilot study when I was at the University of Cambridge, I got some funding when I was there, and this is the foundation of our current project here at UTM. It became clear to me that not only is AI just as good as the human eye, it actually has the potential to be better. So I suspect not only will we be able to develop a deep learning phytolith identification workflow for the Southern Levant, our research aim that helps us build larger data sets, I think it's going to help us build more detailed data sets as well, such as species levels identifications, which we are on the precipice on in my lab already, and likely long-term, more detailed ecological information, such as growing conditions or taphonomic assessment details. So this means that for the past few years, we've been focused on building the infrastructure in my lab that will allow us to achieve our research aim of building a deep learning phytolith identification workflow for my region, Southern Levant. So in brief, what is this foundational research we are aiming to scale up? That is our first algorithm paper, which was published in 2022 with the Journal of Archaeological Science. This is our original algorithm. It is our more recent paper, which is opening the black box of that original algorithm. So we're applying visual explainers to our identifications. This is with Annals of Botany 2025. And then it's also a comparison of our algorithm to humans. And this is a paper that's in review currently with the Journal of Archaeological Science reports. So comparing our algorithm to manual approaches. So back to our foundational research, the first algorithm paper. This is titled Automatic Detection and Classification of Multicell Phytoliths Using Deep Learning-Based Algorithms. So again, I was at the McDonald Institute. I was given a small grant, and I used this to fund a pilot study working with Dr. Hector Arango and his then PhD student, Iban Berganzo-Bezga, who was then at the Catalan Institute for Classical Archaeology. And I wanted to see if we could use deep learning algorithms to identify multicell husk phytoliths. This was important to me because I work in the Near East, and this is where we get some of the best identifications, most specific identifications. They're also very complex. There's multiple diagnostic criteria that we have to use and balance to make IDs. It's a real art form. And in this work, I like to work from the known to the unknown. So I wanted to look at taxa that Arlene Rosen had used for her 1991 publication in Phytolith Systematics. So using my own comparative materials and my personal microscope and camera, which, are very basic, I imaged a few hundred phytolith multicells for each taxa. So Hordeum, Avena, and Triticum. Once we had cleaned up the data and tagged the images, we were closer to only around 100 images for each taxa. And these images were then split into training, validation, and test subsets. The model then pre-processed the images by breaking each of these 2,560 and 1922 times pixel images into 36 tiles. Each individual tile is then classified as either background, so there's nothing there at all, artifact, so there's something there, but it's not a phytolith, or then the phytolith and the taxa, so Hordeum, Triticum, or Avena. So this is at the top there, an image of the workflow. So you have your phytolith image, it gets pre-processed, you have classification, the black box, and then you have the identification, and that is that grid. So again, each of those images are broken into these tiles, and then each of these tiles is identified. So the red grid you see, that's the model's identification, red being Avena, blue being Triticum, and green being Hordeum. So this phytolith was correctly identified as Avena. If there was discrepancy between the tiles, so you could have images that had different colored tiles, what happens is the overall classification is the category of taxa that you see the highest percentage of. So if there are more tiles that are Avena, the image is classified as Avena. We also used confusion matrices. So that's at the bottom right there to help us better understand which taxa categories the model was most proficient at identifying, but also which ones were getting confused for which. But the main issue we had, of course, is there's this black box. But I should say here, the accuracy, the average accuracy we got of 93.68% was frankly surprising to all of us. That's all I can say on that. We were surprised at how effective the AI was at classifying phytoliths. And maybe I shouldn't have been surprised. If we look outside of our discipline, they are using very similar computer vision models, particularly in radiology, for example, identifying cancers and such. So, it was just surprising at how effective actually the algorithms can be. But again, we don't know why they're being effective, and that is a problem. So this leads us to the next stage in our research. So Iban finished his PhD with Hector, and he came with me to Toronto, and he was a postdoc and worked with me for that first year while I was setting up the lab, working on our original training data set and trying to use visual explainers to open up the black box. So this paper recently published with Annals of Botany is called Deep Learning Black Box and pattern recognition using guided GradCam for phytolith identification. So in this paper, we wanted to make it as clear as possible what characteristics the model was using to make identifications. Both to verify that the phytolith identifications were based on the phytoliths themselves and not some sort of background issue in our images, and to help us refine our training. So it's this sort of cyclical thing. We can keep improving our training data. So one of the ways of trying to understand guided GradCam or these heat maps is essentially you can look at this sort of cat versus dog. So in traditional models, if you're not using a visual explainer, it's a black box. We see that at the top. You have cat, it's a cat. You have a dog, it's a dog, but we don't know why. So using these heat maps, we can see the red being what the algorithm is prioritizing or emphasizing versus the blue, which is what it is de-emphasizing. And what this allows you to do is to, one, make sure that you're looking at a cat and a dog, or the characteristics of the phytoliths you're interested in, but it also allows you, if you're getting incorrect identifications, to go back into those individual identifications and say, okay, why is this cat being classified as a dog? What are we looking at? And you can see here in the bottom right-hand corner, it's actually looking at the grass. The grass is what the model was emphasizing. And you can know then, I need to feed it more information, more clean tagged information, telling it that this is a cat. So for us, you can see, I thought a really nice image here is our Avena image, the second one in from the left. And it is really clearly emphasizing the model, the papilla here. And that's a really nice picture. And then the one beside it, you can see that in that hordeum, or the triticum, I'm sorry, it's clearly emphasizing those long cells, the dendritic long cells itself. But we go even further when we use the other visual explainer layers, such as the guided back propagation and the guided GradCam. I think it becomes even clearer. So those two gray images, the right-hand side, but the two images on the left of that segment set, you can see if you look closely, the edges of those phytoliths are really clearly being emphasized by the model. And this is really important because although perhaps not surprisingly, we found that the wave pattern is really key. It's a really key identifying characteristic for the model, confirming Rosen's 1991 paper and years of manual research and intuition, of course. So this is, again, particularly clear when we look at those images. The use of these visual explainers, I think, is absolutely fundamental. I think there is, it's a mistake to use AI if you are not using these visual explainers, because it is really about increasing transparency. That is a real benefit of the AI. It can increase the transparency. Every single ID can have one of these images that people can look at if they choose to. And it ensures that every identification is essentially explained. Now this then leads us to our most recent paper, and this was done with my grad students, Melanie and Lachlan, and here we aim to compare the original algorithm, so again, our original algorithm, to a survey of phytolith experts. And the paper is entitled Experts Against Automation, Comparing Artificial Intelligence and Human Identification of Phytoliths. And this is in the Journal of Archaeological Science Reports. And in this paper, we used SurveyMonkey to build a survey of 18 phytoliths from our original test set. And we were able to solicit 12 survey respondents, which is a small survey size, that's true. But the pool of individuals who have the necessary skills to identify Near Eastern grass husks is small. So we felt this was a reasonable sample size. And the survey was completed over the summer and fall of 2024. We asked the anonymous respondents direct questions about the features they employed in their identifications, how certain or confident they felt, for example, in their identifications, and we timed it. So of the images used in this survey, so of those 18 images, the algorithm classified all of them correctly. So I'll go straight to here so you can see these are those 18 images. They were all identified correctly. If you look at the hordeum, the middle column in green, and we follow it down to the image second from the bottom, that's a really great example of a phytolith that the algorithm found confusing, right? So we have all three different classifiers, classifications present in that phytolith. However, it was still ultimately classified correctly. So the algorithm, I am not telling you the algorithm has a 100% accuracy, but what I am saying to you is of the images that were used in this survey, the algorithm did classify them all correctly. Whereas manual experts only achieved an average accuracy of approximately 44%. Now, again, there are many issues here as far as they were given a picture. When we do identifications, of course, we are working under our microscope. We are going up and down. So it's not the way we would normally work. We also normally don't necessarily identify every single phytolith we come across multi-cell. There are some phytoliths that do not have the features we would use, and we would just say, this is a husk. So there are several things that are impacting these very different numbers. But let me break this down even further. Manual experts struggled most with Avena. They only had a 26% accuracy versus Hordeum, which is 54%, and Triticum of 49%. So we're not suggesting in this paper that this algorithm, that our original algorithm is ready to be used in archaeological contexts and can beat out a manual expert classifying archaeological multi-cell husks. But what we are saying is that the original algorithm is far more accurate, consistent, and quicker than manual experts at identifying between Avena, Hordeum, and Triticum multi-cell husks from cropped images taken from clean comparative slides. So that's a lot of qualifiers, shall I say. But to put some more weight behind this, all of our survey respondents agreed to be authors on this paper and contributed to the editing and preparation of the manuscript, which I said is currently being revised following peer review. So essentially, this survey demonstrates that AI tools, such as our original algorithm, can enhance accuracy and efficiency for specific identification tasks, and they show promise for future archaeological application. But this algorithm is not ready for archaeological application yet. So how do we move from this foundational research to our actual research aim? So a deep learning phytolith identification workflow for the Southern Levant. Well, we need to take away one key point from our foundational research. Algorithms aren't the limiting factor. Training data is. Deep learning models are only as good as the training data they have. So let this sink in. Crap in, crap out. This is something I think we are all going to hear a lot of in the coming AI age. So how do we scale our research? We need to build the infrastructure that allows us to build the best possible training data for our research aim. So when I was hired by U of T, I was given the resources to make this possible: physical lab space, institutional resources, and time. So as a team, we at the RLEA have focused on using these advantages to develop and build 4 infrastructural foci to help us scale our research. That's a lab database. So we worked with the Data Science Institute at the university. This is helping us keep all of our data and metadata associated with every kind of sample that's developed in our lab. So archaeological, training data, or experimental data, for example. And because it's bespoke, there will be opportunities to integrate our database directly with our algorithms and their outputs, so the data, at a future point. Our microscope and our lab facilities, that is Hazim, one of our students who rebuilt our computer. We already are on computer number 2, which is crazy. So we have a Nikon Eclipse NIE microscope paired with a Nikon Digital Sight 10 camera, and this allows us to scan 4 slides at a time, creating high quality images with far more pixels than, and therefore information, than our original algorithm had access to. Training data, we have been ashing and preparing slides constantly. So using my my comparative collections, which were subsampled from UCL's Hillman collection, and from my own collection, the Mount Scopus collection, which was collected by botanists in Israel over 4 seasons. We're also actively soliciting more materials. So if anyone is interested in collaborating with us, please email me. If you have materials from the Near East, particularly grass husks, these algorithms, they are so data hungry. And research team. So we have a team right now, as I've already mentioned, of approximately 30 undergraduates. I think we're even bigger now. We just hired some more AI algorithm students. And as I said, they work in a range of disciplines, and this helps us all achieve our research aim. And this infrastructure as a whole is allowing us to scale up our research, particularly because of our Nikon imaging workflow. So how are we scaling our research? We have an Nikon imaging workflow. This is our imaging system. This allows us to automate taking high resolution images from our slides and how we're building our training data. And in the future, how we will apply our algorithms to archaeology data. This is how we aim to break out of our manual microscopy methods. Then we have our training data labeling app. And this is how we currently have students, tons of students helping us label our training data. This is a major bottleneck. We're building large numbers of images, and all of these images need a person to label them so they can be used effectively as training data for our algorithms. So this is also called pre-processing. Algorithm development. So now that we have our growing training data set, we are about to get our GPU node and server online. We have a team of undergraduate computer science students who are playing with all of our data. And we're working with these students to trial new models, new ways of visualizing our data that are more intuitive to archaeologists, for example. And then we're also doing a bit of sort of blue sky development. So what is the Nikon imaging workflow? On the right, you can see this is our original manual workflow, which was plagued by computer processing constraints, file size time needed and it needed a lot of human oversight. So based on this, we created a semi-autonomous imaging workflow using the Nikon Eclipse microscope paired with our digital sight camera. And this hybrid approach, developed in consultation with Nikon imaging specialists, is streamlining the imaging and annotation process. These advances in the workflow have been made possible because we upgraded our computer. We didn't even have that computer for a year, and we realized it just was not up to snuff. It wasn't going to work. And all of the specs have been maxed out on the workstation, only limited by the GPU compatibility with the Nikon program itself. To further this, a fully automated workflow is almost in the pipeline with Nikon. It's almost ready to go. And that will be 100% novel approach, not used before. So I'm going to go through in a bit more detail what this fully automated process is going to look like here. So first off, we have 4 slides on our microscope, and these are imaged at one time. The full slide scans are taken at 10x magnification, and a Z stack of 11 focal points is EDF'd and stitched. In the fully automated version, this step will be the same. Next, the fully stitched slide scan is scanned for phytoliths. The Nikon AI has been trained. We trained it on both small and large phytoliths. And on this slide, you can see here the locations it has flagged for the 40-time imaging. So the large phytoliths are masked in red and the small phytoliths are masked in green. So previously, this was a manual approach, and someone would sit at the computer and tag each point for further imaging. That's not going to be the case anymore. It will automatically image because we've trained the Nikon specific AI. Next, all of the flagged points are imaged at 40-time magnification with Z stacks of around 30 focal points, and the entire slide will be able to be scanned at once. And once it's finished taking all the Z stack images, the file is converted into an EDF, and each individual 3D image is saved as a TIFF. That is then essentially the end of the Nikon imaging workflow. We then transition into our lab data labeling workflow app. Now, this has been automated by our student, Nicholas Iovu, and this is a particularly important point. The training data needs to be clean and clear as possible, and therefore labeling is critical, but it's a bottleneck. Our student volunteers in the lab are essential for this work. So this is when the images are then tiled and they are tagged for pre-training. There are folders in our drive that are labeled by species. And I have a little video here that Nicholas created that we share with all of our undergraduate students to help them go through this process. I might skip this, but essentially we have an app that allows students to very easily click buttons when they see an image for saying which file it should go into. So is it going into a file for being background or object or sorry, artifact or phytolith. We then further, I'll skip that for now, but I can
Hello
share
and
that
welcome
with you
to the
guys.
Sorting Desktop app introduction. So I'll just be showcasing a little bit of
I'm
the
going a little bit
functionality
long
of
in
it.
time, so
It
I'll
is
skip
subject
this.
to
Sorry, guys. And then the final step is sorting the phytoliths into diagnostic versus non-diagnostic categories. So students then look through the multicell husk phytoliths, and they look for phytoliths that contain diagnostic morphological traits such as wave pattern, pits, or papilla. So we are further cleaning our data. Now, this does introduce inherently a bias towards known morphologies. That is true, but we work from the known to the unknown. So while it's again true that the AI might see things in these phytoliths that we don't see, they work with pixels, it's not the same as us. And we are skewing our training data towards those things that we know for the human eye are identifiable. I think in the long run, this will be nullified because we have tons of images, extensive images of the full plant parts that are going to enable us to mitigate this in the future. And I think it just makes sense to work from the known to the unknown. So this workflow is allowing us to rapidly expand our training data. We currently have about 350,000 tiles. When I asked my students yesterday, this contains 3 classes, again, the phytolith, the background, and the artifact. And this is from 3 classes from 9 genera so far. So we have 9 species of triticum, 4 species of hordeum, 9 species of avena, etc. And we aim to hit approximately 30,000 clear, high-quality tiles that contain diagnostic features for each husk category. Other less complex morphologies as we move forward are less likely to require so many tiles. So, working with this new training data, we have a team of undergraduate computer science students headed by our student Kivan Buck, and they are using this growing training data to trial new algorithms and new ways of visualizing the data, as well as some blue sky development. So we'll see where that goes. And we're focused right now on developing the VGG19 ensemble models. They trialed a sample of this with our some of our new training data, over the summer. And with triticum in particular. And for some of our species of triticum, we have as many as 4,000 tiles already prepared in the summer. And we were hitting accuracies with this ensemble models as high as 99%. I suspect as we bring in more taxa, these accuracies will adjust. They will not be as high. But the more training data we have with these powerful models and the compute we're now having in our lab, it's really promising. We're also working on new ways of visualizing the data. So to show us how the algorithm is making its predictions, and this includes UMaps, which you see right there. And this is essentially almost like nearest neighbor stuff, right? We're seeing which categories of itch. So imagine each of these colors was a species of phytolith, and you can sort of see how specific the identifications are, where the confusions are, how related each of these are as far as their morphology. I think this is going to be a really powerful way when we start looking at species level identifications, that data. But we're also going to, of course, continue using confusion matrices and GradCam heat maps, which are going to help us be sure about the identifications that our models are making. And some of the blue sky research we're investigating includes synthetic data generation, which we think is probably going to be critical to our model into allowing it to do single cell identifications effectively and quickly. We're also interested in unsupervised learning, which with enough training data and compute might be a more nimble approach to expand our AI tools to multiple regions. But I think computer science, the AI movement in general, actually is moving towards away a bit from the unsupervised learning right now and focusing more on really clean, well-labeled training data. So we might be here for quite a while doing a lot of labeling. But if we're getting accuracy rates as high as 90, if we're in the 90s, it's amazing, right? Let alone 99. So next steps. We will be applying our new models and our new training data towards species level identifications. We're focused on triticum in particular right now on doing all that imaging. We then are going to be expanding out, of course, doing far more species of wild grass husks in the Near East. We then want to turn our attention to archaeological samples. So how do we integrate archaeological samples in with this model? And in particular, one of the big questions we want to look at is domestic versus wild triticum. Then we're going to continue working through our taxa in the Near East. I think Cypraceae, sedge cones will be a really exciting area to go into, Quercus, etc. And we're just going to keep working through these taxa from the more economically important species, the more diagnostic species, to the less economically important and the less diagnostic species until we reach our aim, which is, of course, a deep learning model that is fully automated for this region. Now, long term, in the lab, we aim to make this workflow from multiple regions and then make that data available easily, hopefully through something like a prehistoric plant atlas. So you have a picture here. This is like a Google map, but imagine you could have something like this for archaeology sites, and you could click it and you could see the different plants, the data, etc. I think that would be a really exciting future plan. Again, very long term, because as you can see, we've already spent years just trying to build training data from one region. So again, as has been made probably abundantly clear to you, this work is resource intensive. It uses people, infrastructure, time, and energy. University grants will help, but grants aren't guaranteed. And honestly, in my experience, I find that reviewers right now are still really hesitant when it comes to AI projects. I think just the topic is really charged. And I suspect one of the ways that we can keep developing our training data and our algorithms is going to be through an accelerator type model. So we think about, the Oxford accelerator model. Maybe we're going to have to do something like this with consulting as well, just to guarantee continual resources coming in so we can keep developing our tools and our training data. Now, I realize after all of this, sorry, I've gone a little long, as members of the International Committee on Open Phytolith Science, you were all likely very interested to know how my lab and I plan to navigate open access science. I think that the use of AI tools has the potential to make our work more transparent and therefore also more easily open access. But the question really is, how open is open? So ICOPS advocates, of course, for open data, open methods, and open software. So I think this is really achievable with AI-powered research for sure. I think the application of AI tools, particularly the algorithms we're employing, can help our lab achieve open data, open methods, and open source algorithms. Our lab is committed to making our data, the data that underlies all of our papers, open access using repositories like Zenodo and GitHub for all of our papers, actually. And any algorithms we employ, once published, they will be available on GitHub, open access. And also examples from our training data. We will also make examples from our training data open access. So the full images will be open access, some of them. But our ability to make all of our training data open access is more unlikely. And that is a topic that requires just more thought in general. Just again, it is so resource intensive to do. And we need to have a little bit more of a think of how we keep our lab afloat, how we keep building these tools, and what we need to make that happen. And with that, I will end it there. I know you will have many more questions, or I hope you will. And thank you for having me here. And thank you to my team and all the funding bodies that we have, of course. And yes, thank you, Jennifer, for inviting me.
Thank you for that fantastic talk. I'm going to open it up to questions, but I want to actually start with one quickly first, coming back to that very final point you have about open data and open access. To kick off this sort of questions and answers and discussion, and ask, you say that sort of like data can be published alongside this, and I'm wondering if you've been thinking about, a bit like we have like the raw R code that has to be published, or the DNA reads that have to be published. You mentioned algorithms have to be published, but do other things, do you think maybe to do with AI, have to be published, like some of the black box decisions? Those
100%.
image checkings need to be published, and how would you do that beyond putting it just into a repository? Like, how would you explain the methods so that people could replicate it?
Well, I think that is, and Melanie and Lachlan, please at any time at this point now, if you want to jump in, feel welcome to do that. This is the thing. So I don't know that this is a real team science approach. So is there a way to make it so that anyone with very little computer science background can just go and apply the algorithms and understand how they're working? I'm not 100% certain. But even those, the black box layers in our algorithm, all of that code, we publish it. So in the Annals of Botany, that's all available on GitHub. So anyone can take it. And what we will be doing also is making sure all the images are available as well. But I think there's also a place, and I'm not 100% certain. I think it's probably, so we did the database, right? And my hope is eventually that all of these images will be saved in our database, but they'll also be saved in one of the many cloud programs we're using. I mean, the amount of cloud programs we use right now in our lab is crazy. So AWS, let's say. So perhaps it's as simple as having files available on AWS for anyone to download, whereby you have all of the Phytolith images that have been identified, the plain images, and then you also have them with the visual explainers. So you could have a file for visual explainers. I'm not 100% certain how we could make it super easy to like scroll through, let's say. But at least if we can make it all available for now, and who knows in the future, perhaps someone else has some interesting program ideas for this. But it is, you have the problem that it's going to be a lot of data. And sometimes, in being open access, you can dump so much data that it's almost not open anymore, or it's not easily maneuverable, shall we say. So that will be a balance that we have to try to strike, I guess, as well.
Yeah, absolutely. I think it's going to be an interesting sort of seeing how the interaction between AI and open access develops.
Yeah.
If anyone has any sort of questions that they want to ask, you can use the raise hand icon. Or if you don't want to sort of like, unmute and speak, you can drop it into the chat box and I'm happy to read it out. So does anyone have anything they want to sort of bring up and discuss with this really awesome topic? Scrolling through to see if there are any hands. Okay, while people are thinking, then I'm going to ask another one, which is how much, because I find this really fascinating. How much, so you mentioned that this idea that this is very sort of at the moment focused on the southern Levant.
Yes.
How difficult do you think it would be, like focusing very specifically on the wheat and the barley and the oats, which are the current focus? How difficult do you think it's going to be to take those with the current algorithms that are being used and apply it in, say, another area, given ecotype variation, given environmental differences, maybe even like time difference and
So
domesticatedness?
I think we're going to have a very specific regional models. I think we can accommodate the sort of, like when we're dealing with plants that are sort of domesticated or not domesticated, I think we'll be okay sort of being in one region for that. But I think every region, and how do we define region? We're going to have to make those arguments at the time, is going to need its own training data set, its own appropriate images. I also think you're right, sort of, I think so again, we work from the known to the unknown. We know there are papers out there that suggest water, so precipitation can impact silicification. So this is going to be something I suspect, therefore, that the AI can also pick up on. And I have a really great, I'm working with an ecologist at Western right now. She has an amazing phytolith collection that she collected years ago in North America from like 14 different locations where she meticulously recorded the growing environment of these particular North American grass taxa. So we're going to try to see if we can pick up different growing conditions using this. I think we also need to move into growing experiments. So growing wheat, let's say, in a greenhouse with differing varying levels of maybe CO2 and water. I mean, the amount of variables we can be testing is almost mind-boggling, because I do think you're right, it will impact the application. And then we're just going to have to adjust and pivot. And if we realize that growing conditions, let's say, the algorithm isn't very effective at identifying grass husks from the last glacial maximum, well, then we might need to pivot and reconsider how we build more appropriate training data for that time period. You're right, we're going to have to be thinking about all these things as we move forward. But also, I should say, regionally speaking, Melanie, my PhD student, she works in Mesoamerica. So she is also simultaneously, at the same time as we're building our training data for the Near East, she is also imaging training data for Mesoamerica. And she can speak more to this for her materials, because of course she's focused on very different types of phytoliths that she's interested in looking at for Mesoamerica. But we're focused on totally different types of phytoliths there. For example, more Ericaceae and etc.
Yeah, I think
Yeah,
if I
super cool.
Oh, go ahead.
This is Lachlan, please, and Melanie, it's the below there.
just to add to that, like the actual framework of AI is relatively straightforward. It's training, validation, and then a test. So like Monica was saying, it really comes down to what is the training data you're feeding it. So if you're in a different region, the algorithm is really straightforward. If you have, let's say, 500 images of millet, then you could really start training an AI algorithm to identify millet. It really just comes down to like what sort of metrics and other characteristics do you want to look at in the archaeological record, and then finding that in your training data. So if it's like Monica was saying, growing conditions, or if it's different plant parts or wild versus domestic, all of those controls can kind of be controlled for and then trained in the algorithm.
Yeah, so like also thinking about like different regions and different types of phytoliths. Once we have like the main algorithm, like we have the multi-cell, it just takes a bunch of like fine tuning as we add more like classes and more species, and that's pretty much it. Like we're not rewriting the whole algorithm, so it should be pretty streamlined from there.
I think that comes back to my sort of question about the open access. If the algorithm can be kind of like not perfected, but you know what I mean, like sort of like streamlined to a set of, and again, I don't want to use the words like sort of idiot's guide to AI, kind of like plug it in and see where you go. If it could kind of be like sort of published in a way that was like, like here's how to do it, literally like go out and get at least this amount of raw images that are in this format. Here is the algorithm. You're going to need to tweak this factor, and this factor. Could theoretically somebody, I don't know, let's say working in Korea on stuff from the Chulman period, where you've got very few grasses, but quite a lot of, oak and chestnut and hazelnut, theoretically plug in the reference collection images and run it and then compare it to archaeology, theoretically.
Theoretically, yes, exactly. You could do that. And I think this is one of those things, like this is a new technology and most of us aren't computer scientists, but it is going to be normalized. Like it is going to start being more normal. And I think exactly as you said, like you think, okay, do we need this sort of like a guidebook? But I think those guidebooks actually exist. Do you know what I mean? Like if we go out and look, those papers, maybe not published by archaeologists, but they are out there. And again, there are going to be models that are better, and there are going to be models that are worse, right? It comes back to the image with the crap in and crap out. And like Lachlan said, people just are going to need to make sure that they are properly testing and validating their models and making sure that they have the appropriate training data if they want to be applying it to the archaeological record.
Kind of in the same vein, the models we're using are pre-trained, so we're not making them from scratch. We're taking these models that have already been made and we're just fine-tuning it and training it with our data. So it's something that people can do, and they're open access pre-trained models that you can use.
Thank you. I'll add it to the to-do list. Does anyone else have any questions that they would like to ask, so it's not just me sort of dominating the conversation?
I'll go. I mean, hi. It's not really a question. It's more like, first of all, it's an offer. We've got lots of millets, like experimentally grown as well. I think that Francesca is somewhere in the audience, so you know we have plenty of material if we want to try and do some tests. Also, I'm trying to understand whether the growing condition, whether can be used, whether the model can be used for that as well. I want to maybe do a little step back, because the other thing that was really very impressive for me in this talk was the automatic scanning and imaging of the slides. And can you tell us a little bit more about that? In the sense, is that something that you know, other laboratories can get, like, you know, that we can buy from Nikon or from, I don't know, something like that? Because that sounds also really, really amazing.
I'm going to pass that off to Melanie. This has been something that Melanie and I, we said we need to do this, and she has just spearheaded working with Nikon directly. So please, Melanie.
Yeah, so it's an AI program that you can get from Nikon, and it comes with the microscope that Monica got in the program.
Okay.
So we had an undergrad who spent, let me think, maybe I want to say like 2-3 weeks masking all those spots. So spots that she identified as like large phytoliths and spots that she identified as small phytoliths. So you could go in and do that. And then based on all that training she did, it's now able to identify the phytoliths on its own. So it's definitely something that you can do as long as you get that program and pay for that AI that comes in there. And then
Now, you're
Nikon has been
making it
really
sound
good.
very easy, Melanie. It
Okay,
wasn't
no.
as easy as
Yeah.
you're suggesting. We struggled a lot with compute.
Yeah, okay, so that's so training the Nikon AI specifically isn't crazy. It's understanding like file size, how much computer like compute power you need, and then what you actually want to take photos of. So for like the multi-cell phytoliths, it's a bit easier because they're so big. But stuff when we move over to like more single cell stuff, that might be a little harder to pinpoint on the slides.
Yeah. Fantastic.
Awesome.
Yeah.
I
That's
should
awesome.
also add to that, you know, we spent months and months working with a computer that was not fit for purpose and trying to like set it to go and image overnight, and then it would, you know, hit 50 phytoliths and die. And
Yeah.
I'm sure it was driving
Yes.
me absolutely.
Yeah, so I would set
Every
it
time I
to
talked to her, I'd be like, how many
Yeah.
phytoliths have we done now? And she'd be like, and we
Yeah.
finally just trusted one of our students. We had a student who he's a biology student, but he's actually just really on top of computers. And he said, listen, I could make you a better computer, because I was speaking to Nikon and they were quoting me like $12,000 for the computer that we needed. And he was like, no, I can make this for like 5
Wow.
and even better. And he did. And we took a risk, but I'm glad we did. He did a
Yeah.
great job.
Yeah. And are all these specifications somewhere in the papers, or can we maybe send you an email and ask you for a little bit of guidance, whether, you know, in case we are thinking of, you know, setting up something? I mean, obviously not what you guys have, because you've been working on this a lot, but maybe, you know, starting.
So Melanie is working on, so this paper in particular, the workflow paper, is going to be one of Melanie's chapters for her PhD.
Okay.
So that is currently in the works, and those details will definitely be available there.
Awesome.
But Carla, you can email me, and me and Melanie can share with you that, you know, where we're at with that right now. No problem.
Fantastic. Thank you.
Perfect. That was actually going to be my closing question for this. As we are very close to time, I was going to say, could you drop us a link with the microscope and computer details, and we could pop it round the mailing list. But I think it's probably better if people are interested to directly contact you to get the details and to have a chat about it. So I
Yeah,
think
and
we're
I
going to
should
wrap up
say,
there. Oh,
I know that thank you for giving us the opportunity to share where we're at, because we are going to be publishing way more in the coming years. But like I said, it's like it's just been trying to build from the ground up and knowing like if our end aim is this, then we need to build this in the meantime. And it just has taken time, but hopefully, you know, the workflow paper is going to come out soon, and our new training data and our triticum species level ID paper, and it will just keep rolling from there.
absolutely, and it's really exciting to see this work happening. And I would say that if people are really interested in this, which I'm sure they are, and they have questions that they didn't want to ask right now, they can drop you guys an email to ask sort of in private. And I will say thank you once again to Dr. Ramsey and her students that are here, Lachlan and Melanie. Thank you so much for joining us and for being part of this conversation. And to say that next month, so in November, we have our next speaker, Dr. Ethel Alway. I think I'm pronouncing her name correctly. The researcher at the Catalan Institute of Human Paleoecology and Social Evolution, who will talk about unlocking the archive, digital challenges in 30 years of anthropological research. And that will be on, let me get the date up because my screen is scrolled. That will be on the 28th of November from 2 to 3 p.m. UTC, and the registration link is up on the chat right now, but you will also get it in the mailing list that will go around. And with that, I will say once again, thank you so much for what a really interesting talk, and we look forward to seeing many papers to come.
Yes. Thank you.
Thank you,
everyone.
Day or evening or night, depending on where you are.
Good morning, good afternoon, good evening, and good night.
Bye-bye.
I will let everyone leave, and then I will